Hello everyone and welcome to our new lesson on EMSAT Achieve Biology. We are continuing with the same section, which is section 1. Today's topic is the third subsection, which is explain the role and effect of enzymes and other factors like pH and temperature in the chemical reactions carried out in living organisms. We are going to deal with this topic in a very precise manner, just focusing on the key points. So the first thing that you have to know in this is what are enzymes. Now we just had a lesson on macromolecules and now we know that enzymes are proteins. What's the function of enzymes? The enzymes, they are biological catalysts. Catalyst is anything which speeds up a chemical reaction. When I say biological catalyst, I mean they speed up the reaction in your body, in the living organisms, right? And you have to understand that enzymes, they are very specific in action. So every enzyme has a particular substrate or one or more substrate on which it will act, right? We'll get to that a little later. So you have to understand when I talk about catalyzation, you have to know that they catalyze very specific reaction. That means they make the reaction happen faster than they would otherwise. And now coming back to the concept of a substrate. So we know what a chemical reaction is. So when on the left side you have the reactants, they combine together to form something called a product, which is on the right side of the reaction, chemical reaction. So enzymes, what do? They act on a reactant, which is a substrate molecule. It can be one, it can be more. So this is known as the enzyme substrate. Okay. And so here you have, for example, an enzyme. So enzyme have different pockets, okay, and the pockets are of different shape, which can accommodate different types of substrate. For example, this enzyme has two substrate, A and B. And for example, this one is in the shape of a rectangle. So reactant A can, or the substrate A can come and bind to this. And B is an inverted triangle, so it can come and bind to this active site. So the site where the substrate will come and bind in the enzyme is called the active site. This is a very important concept. Now the next thing you have to know are the two key models that is asked in most of the questions related to enzymes for your EMSAT biology. So the first thing is lock and key. So it's a very simple model. It's an earlier theory. And it says that, okay, every enzyme has a particular shape which can accommodate the substrate with that particular shape. For example, this enzyme has a circular shape, a semicircle thing, and this substrate is of circular shape. It can go and bind and find an enzyme substrate complex, later on making a product. What is the induced fit model? This model says that the enzyme has a specific shape. They are not flexible. The active site is not flexible. So if it's triangular, a triangular substrate can bind. If it's circular, a circular substrate can bind. But induced fit model is a model which talks about flexibility of the enzyme. It says, okay, my enzyme has this shape, okay, but the substrate is circular in shape. But this substrate belongs to this enzyme. So what will the enzyme do? The enzyme will change the shape of the active site in a way that now it can bind to that circular substrate. So look at the shape that we had before and how the enzyme is changing the active site in a way that now this substrate can come and easily bind to it. It's called the induced fit model, and it's one of the most widely accepted theory for enzymes. Okay. The next one they ask you every time is about the activation energy. What is activation energy? Let's go into that first. Activation energy is the amount of energy that you need to begin a chemical reaction. As simple as that. So if the activation energy is, say, 20 kilocalories, so I need at least 20 kilocalories of energy to start a chemical reaction. Anything less than that will not start that chemical reaction. Okay. What does the enzyme do? The enzyme will lower the reactions, so activation energy. So here, for example, this was the initial activation energy. I needed this much amount of energy to start my reaction. Now, when I add the enzyme, look how the activation energy is coming down. So now I don't need so much of energy now. I just need this much of energy to start my reaction. This is what the enzymes do. They lower or they decrease the activation energy of your reaction. Okay. The next thing is the factors which affects the enzyme activity. The first important factor is your temperature. Temperature has a big influence on the enzyme activity. Remember that. Why? Because the more the temperature, the more faster will be the movement of your enzymes. Okay. For example, the more heat means more kinetic energy, right? More fast the enzymes will attack each other, will collide with one another. So more the temperature, more will be the rate of reaction till a point, which is known as the optimum temperature. Every enzyme has an optimum temperature where the activity will be maximum. After that, even if you give more temperature, the activity will not increase anymore. In fact, it will decrease. This is the temperature where denaturation takes place. Denaturation is when you're destroying. Remember, enzyme is a protein. So when you heat it too much, you're breaking the peptide bonds, you're destroying the structure of the protein, you're destroying your enzymes. This is what is known as denaturation. So at very high temperatures, the enzymes gets denatured or destroyed. Okay, they can no longer work. They're of no use to you after that. Okay. Next, coming to pH. So every enzyme, just like they have an optimum temperature, they also have an optimum pH. Okay. That means that at that pH, it will work the most efficiently. So anything higher or lower can affect. So if it's low, the effect will not be that good. If the pH is high, also the activity will decrease. Okay. Now, enzyme concentration. The more you add enzymes, the more faster will be the rate of reaction. But remember, the rate of reaction will be faster the more enzymes you add, but till a particular level. Once you have, for example, you have 100 substrates in your reaction and all the 100 substrates are used. And after that, if you even try to add more enzyme, nothing will happen because you have exhausted your substrates. So it will go to a particular point. After that, once all your substrates are used, even if you add more enzymes, nothing will happen to the rate of reaction. Okay. Coming to the substrate concentration, of course, if it's the same concept as increasing your enzyme. So you can give substrate, the rate of reaction will increase up to a point. After that, so if you have 100 enzymes and you have 200 substrate and you have used all your 100 enzymes, even if you add more substrate, nothing will happen to the reaction because you have already used all your enzymes. So the concept of the substrate concentration and the enzyme concentration is almost the same. Okay. Now let's go on to practice some EMSAT biology questions. So the first question says, increasing the temperature above the optimum for the enzyme results in loss of enzyme activity. How is this explained? We know why the enzyme activity is lost. It's everything related to denaturation, right? Let's look at the different options that we have here. Less frequent collisions between the enzyme and the substrate. It's not going to happen. Why? Because when you increase the temperature, the collisions will be more, not less, right? Because we are increasing the kinetic energy. Reduced kinetic energy? No, we just said increasing the temperature means increasing the kinetic energy. Now coming to this one, substrate molecules move faster. Yes, they do. And effective collisions are likely. So these are completely opposite statements. When the substrate molecules are moving faster, how can the collisions be less effective? Okay, there will be more collisions, of course. Now, the last one. The shape of the active site is changed and the substrate will no longer fit into it. And that looks one of the correct answers because, of course, if you are heating it too much, the shape of the active site will change. The enzyme will get denatured and the substrate can no longer fit into it. So your correct option would be option D. Remember, I've told you, always go by the process of elimination. Look at every options that you have and try to figure out. If you think this is one of the wrong answers, quickly just eliminate that answer. Whatever is left, try to figure out your correct answer from the remaining options. Okay, coming to the next one. Which of the following statements is correct about the enzyme substrate complex during an enzymatic chemical reaction? It cannot be affected by the presence of other molecules. It dissociates once the reaction terminates. It is permanent form of the enzyme reaction, or it occurs independently of the enzyme's active site. First of all, it cannot be affected is wrong. We know that there are many factors that affect the enzyme's activity, right? Okay, let's look at the other options here. It is permanent form. So enzymes are never permanent. Like if enzyme is something that is recyclable, you can use the same enzyme over and over again, because enzyme have this quality. Once they come to your reaction, they catalyze it and they go back the way they was. Okay, so you can recycle them. They're not permanent. Now, it occurs independently of the enzyme's active site. Actually, no, because enzyme's active site is actually where the substrate will come and bind. The only thing here is dissociate once the reaction terminates. Yes, I just said they are recyclable. Once their job is done, they will go away from the active site, come back again when you need them. So the correct option here is option B. Coming to the last question, which is which of the following statement is correct regarding the induced fit model and enzyme substrate interaction? Remember, induced fit model, where you change the enzyme active site in a way that it can accommodate the substrate. Okay, let's see the options that we have. Let's start from number D. The structure of the active site is changed by the substrate. Well, we will see to that. The shape of the enzyme is changed by the chemical reaction. Not really. The chemical reaction cannot change the shape of the enzyme. The enzyme directly binds to the substrate to form an enzyme substrate complex. We know in induced fit model, the enzyme cannot directly bind with it because there is a difference in the shape of the active site and the substrate. The active site of the enzyme changes its shape to accept a substrate. Well, the active site of an enzyme does change its shape to accept a substrate. But look at the option A and option D. They're a little confusing here. So here, the active site of the enzyme changes its shape. They will not change in shape themselves. The substrate has to do something to change the shape. You get my point? A and D are a little bit confusing. Here they say the active site itself changes the shape without the assistance of the substrate, which is not true. The structure of the active site is changed for sure, but this change because of the substrate is changed by the substrate. So your answer in this case would be on option D, which is the structure of the active site of the enzyme is changed by the substrate. With this, we finish this subsection. In the next video, we'll be discussing about the cellular respiration and the various other reactions like photosynthesis, which is one of the most difficult topics for EMSAT biology exam. But we will see how we can do it. Okay? That's all for today's lesson. Please subscribe and give your feedback, give your comments, what can be improved. And this will also motivate me to post more videos for you. Thank you so much.